All right, let's dive right into this explainer. Did you know that if you live in the United States, upwards of 70 to 80% of the processed foods you eat likely contain at least one genetically modified ingredient? It's honestly a staggering reality. Yet the conversation around our food systems is often incredibly loud, highly polarized, and, let's be real, a little confusing. So today, we're going to bypass all that noise. We're relying on a synthesis of major 2024 and 2025 reports to separate the foundational science from the speculation. To truly understand the GMO debate, we need to focus directly on how genetic modification changes our crops on a highly practical cellular level. This is the core focus of our journey today. We aren't just going to talk about abstract concepts, we're diving deep into the microscopic adjustments made to a plant's genetic code. We want to understand the exact mechanics of these DMA alterations, how they physically change the biology of the plant, and map out exactly how those precise changes ripple outward to impact our global food supply. Here is our roadmap for today. We'll start with what exactly GMOs are, then look at how crops are changed, weigh the global benefits, evaluate valid concerns and risks, and finally explore different societal perspectives. Okay, Section 1. What exactly are GMOs? Let's define genetic modification. For thousands of years, humans have used traditional breeding to change crops, but that process involved blindly mixing thousands of genes together and waiting entire generations through trial and error just to see if a desired trait emerged. Modern genetic engineering, it's entirely different. Scientists are essentially using molecular scissors. They can isolate a single specific gene responsible for a beneficial trait, like resisting a disease, and introduce, remove, or edit that exact genetic sequence within a plant's DNA. It is a highly targeted, surgical process at the cellular level that alters the fundamental instructions the plant uses to grow. The agricultural sector is currently facing a truly unprecedented hurdle, trying to feed a rapidly expanding global population while dealing with the unpredictable extremes of climate change. So the modifications being made in labs today aren't random experiments. They are engineered specifically as survival mechanisms for our food supply. By targeting these 4 specific areas, improving yield, boosting nutrition, increasing pest resistance, and building climate resilience, scientists are attempting to design crops that can withstand severe droughts, fight off invasive insects, and produce significantly more nutrient-dense food, all without requiring us to clear millions of acres of new farmland. Moving on to Section 2, how crops are actually changed. Let's look at some real-world transformations. Historically, insect pests like the European corn borer literally destroyed massive portions of global corn harvests by eating the plant from the inside out. To solve this, scientists found a naturally occurring soil bacterium called Bacillus thuringiensis, or BT. They took a specific gene from this bacterium and inserted it directly into the corn's DNA. Now, the corn itself produces a protein that is completely harmless to humans because of our highly acidic stomachs, but absolutely fatal to the alkaline digestive systems of those specific boring insects. When you compare robust, engineered BT corn to a devastated, hollowed-out traditional hybrid, the difference is stark. The crop is literally protecting itself. Now think about this. Vitamin A deficiency is a severe public health crisis, causing preventable blindness and even death in up to 500,000 children every year, particularly in regions where rice is the primary staple food. To combat this, golden rice was created. Scientists engineered the plant's DNA to biosynthesize beta-carotene, which our bodies convert into vitamin A, right inside the edible part of the grain itself. That single genetic edit transforms the grain's biological makeup, resulting in its distinct golden color. It's an elegant, sustainable dietary solution designed for vulnerable populations where traditional vitamin drops simply can't reach everyone. Here's a wild story. Back in the 1990s, Hawaii's entire papaya industry was on the absolute brink of total collapse due to a devastating pathogen known as the ring spot virus. Traditional treatments were completely failing, so in a brilliant feat of genetic engineering, scientists took a harmless piece of the virus's own genetic code and inserted it into the papaya's DNA. This effectively acted like a vaccine, inoculating the plant and allowing it to recognize and fight off the virus. It worked so incredibly well that today, more than 80% of Hawaiian papaya grown is genetically engineered. It's a stunning example of how targeted DNA modification successfully saved an entire agricultural industry from extinction. Weed management is an enormous, labor-intensive burden globally. weeds compete with crops for sunlight, water, and soil nutrients, which drastically reduces harvests and ultimately drives up food prices for all of us. To simplify this, soybeans are often engineered to tolerate specific herbicides. By tweaking just a specific enzyme in the soybean's DNA, farmers can apply broad weed control across massive fields. The engineered crop safely metabolizes the chemical and survives, while the competing weeds die off. This completely changes the economics of farming, vastly simplifying management, lowering labor costs, and protecting crop yields on a massive scale. All right, Section 3, weighing the global benefits and looking at the impacts on a macro scale. When you apply these targeted DNA modifications across millions of acres, the cumulative effects are profound. Because plants are explicitly engineered to survive diseases, endure droughts, and face less weed competition, a much larger percentage of what is planted actually makes it to the harvest stage. In practice, this means generating millions of tons of extra food per acre without having to clear natural forests to create new farmland. This dense, highly stable food supply is a really critical pillar for managing global food security in the 21st century. Furthermore, traits like the built-in pest resistance we just saw in Bt corn significantly lower the need for external chemical insecticide spraying. Because the plant's altered DNA acts as its own internal shield, farmers are spared from spraying thousands of gallons of synthetic chemicals over their fields. This doesn't just lower the farmer's operational costs, it translates to drastically less chemical residue running off into local waterways, which protects surrounding ecosystems and aquatic life from unintended toxic exposure. Let's shift gears to Section 4, valid concerns and risks. We have to evaluate the drawbacks. Because obviously, implementing modified DNA into open agricultural environments brings some really complex challenges. Environmental groups point out that an over-reliance on herbicide-tolerant crops actually accelerates the evolution of herbicide-resistant weeds. Think of it exactly like antibiotic resistance in humans. If you constantly expose bacteria to the same antibiotic, the strongest ones survive and multiply. In farming, because herbicides are sprayed more freely, weeds face immense evolutionary pressure. Over time, this accelerates the rise of aggressive superweeds that survive the standard chemicals, unfortunately forcing farmers to revert to even harsher, more toxic methods to clear their fields. There are also profound economic consequences to consider. For thousands of years, the ancient cycle of farming involved harvesting a crop and saving some of those seeds to plant again the next year. But because engineering DNA is an incredibly expensive, proprietary process, biotechnology companies patent their modified seeds as intellectual property. This has fundamentally disrupted that ancient cycle. Today, farmers often legally cannot save these seeds from year to year. They're required to repurchase them every single season. This dramatically shifts the balance of economic power, raising operational costs and creating a stark dependency on just a few massive consolidated corporate suppliers. When we map all of this out, it's like a giant puzzle, and a very distinct balancing act emerges. On one hand, we have profound environmental and health benefits, like drought resistance and life-saving biofortification. On the other hand, we face complex socioeconomic hurdles, like corporate seed monopolies and the rise of resistant superweeds. Based on recent scientific reviews, it's fascinating to note that most of the modern debate centers heavily around these environmental and economic issues, rather than proven, immediate human health risks. Still, there are ongoing, vital calls for transparent labeling and long-term ecological study to ensure the whole system remains safe. Which brings us to Section 5, different societal perspectives. Let's see how various stakeholders actually view GMOs. Well, the broad consensus across the 2024 and 2025 scientific literature is that approved genetically modified crops are safe to eat, and the physical act of altering plant DNA is tightly regulated. However, scientists strongly advocate for continued long-term monitoring of open environments. They want to meticulously track exactly what happens when these engineered plants interact with complex wild ecosystems over decades, looking closely for things like unintended cross-pollination with wild plant relatives to ensure the modifications don't escape into wild nature. Meanwhile, environmental groups highlight a massive structural vulnerability, and that's monoculture. Monoculture is the practice of planting millions of acres with the exact same genetic clone. Their perspective is deeply rooted in the interconnectedness of nature. Even if a specific genetic modification is perfectly safe on its own, wiping out natural genetic diversity to plant millions of identical patented crops is incredibly risky. If a single, new, mutated pathogen comes along that this specific clone is vulnerable to, it could theoretically wipe out our entire food supply. This is exactly why environmentalists relentlessly champion biological diversity. And ultimately, consumers rightfully prioritize transparency. Think about the last time you turned a package around in the grocery store aisle. You probably looked for a bioengineered food label or a non-GMO project stamp, right? While the science might point toward higher yields and robust safety profiles, people fundamentally want to know exactly what is in their food and exactly how it was produced. This undeniable everyday demand for transparency is what continues to drive sweeping labeling laws and ethical governance all around the world. Decisions about our food shouldn't rely on any single dimension alone. We've just explored exactly how altering crop DNA at the cellular level can save entire industries, boost global nutrition, and secure massive harvests. But, as we've seen, we've also unpacked the very real economic dependencies and ecological trade-offs that come along for the ride. So, the next time you're standing in the grocery store, armed with this structured understanding of the deep science and the broader debate, how will these realities shape the food choices you bring to your own kitchen table?